So I took a look at the previous talks, and there were a few talks on sex differences in circadian and sleep-wake regulation, which is kind of nice. So everybody is familiar with a lot of the concepts that I'm going to talk about today. Basically, what I'm going to do in my talk is to focus on sex differences in the circadian and sleep-wake dependent regulation in behavior, with a focus on cognition. And for this, I am going to present data from a specific circadian protocol that I was involved in. Of course, there are many different ways in which you can collect this data in human beings. But this is a comprehensive data set that I have, and I think it nicely illustrates some of the points that I would like to make in this talk. Right. So there is a temporal organization in physiology and behavior, and that is not a surprise for everyone. And it occurs at large time scales. For instance, you have circannual or seasonal rhythms. You have circatidal rhythms, which are periods of 12 hours. And you can see this in the feeding behavior of crabs, for example. You have ultradian rhythms, and these are short rhythms, periods in milliseconds, less than 24 hours. You can see this in your heart rhythm. You can see this in the microrhythms, in neural activity. And then you have the circadian rhythms, which are daily rhythms that are approximately 24 hours in length. And circadian rhythms are going to be the focus of my talk today. So circadian rhythms, as we all know, are generated by a set of core clock genes, and they are synchronized by neuroendocrine pathways originating from a master clock in the hypothalamus, which is the suprachiasmatic nuclei. Now, a very observable and prominent circadian rhythm that we all go through is the sleep-wake rhythm, and this is a behavioral rhythm in light and darkness. Humans sleep during the night and keep awake during the day, and then you have nocturnal animals like rats, which keep awake during the night and sleep during the day. Regardless, the mechanisms of the circadian rhythmicity in this rhythm is the same, and they are orchestrated by core clock genes. Now, in humans, the sleep-wake cycle is regulated by the master clock, which keeps internal time, and ideally sleep occurs or should occur at about the same time every day for all of us. There is also a sleep drive or sleep homeostat, which tracks sleep need, and this is what consolidates sleep and wake. Now, together, the circadian rhythms, the circadian system and the sleep-wake homeostat interact to generate a daily rhythm of deterioration and recovery in behavior or cognition. And this is what we are going to talk about or focus on in the talk today. And in this cartoon figure, I have tried to illustrate the interaction between these two forces. So on the top here, you can see as sleep pressure increases, as the sleep debt increases, you start to fall asleep, and this happens during the day. So with every waking minute, you start to feel sleepier and sleepier, and then somewhere at night, you fall asleep and you recover the sleep debt. Of course, the question is, wow, if I start to feel sleepy with every waking minute of the passing day, how do I keep awake really through the 16 hours of the waking day? I don't really seem to feel very sleepy. And of course, this is beautifully counteracted by the circadian signal, which is shown in the bottom. The circadian rhythm in alertness starts to increase when you wake up, and it keeps going up and reaches a peak somewhere in the afternoon, evening hours. And thereafter, it starts to fall down, reaching a nadir during the early morning hours. So the circadian rhythm in alertness counteracts the sleep drive, which then allows you to consolidate wakefulness during the day and sleep at night. Now, taking the circadian system into consideration, the contribution of this system to cognition is twofold. First, it regulates the structure and timing of sleep, as I have just discussed, in a way that the deterioration and recovery in your cognitive capacity occurs during the day and night, respectively. Now, independent of this, it also directly contributes to cognition, and we can usually observe this when the sleep-wake cycle is uncoupled from the circadian rhythms, such as melatonin. And this is a more subtle aspect of it. Now, in the real world, instances where you experience this uncoupling of the circadian rhythmicity from the sleep-wake cycle is during shift work, as I show in this picture. What happens typically when you are engaged in shift work, especially during the night shift, you're trying to sleep during the day when your circadian rhythm of alertness is going up, and you're trying to keep awake and work at night when the rhythm of alertness is going down. In other words, you have decoupled your sleep-wake cycle from the circadian rhythm of alertness. Another example that I'm sure all of us have experienced is when you engage in transmeridian travel, such as during jet lag. So regardless of whether you travel east or west, what ends up happening is you travel at the wrong time of the day, you arrive at the destination, which is at a different time zone, and you are either forced to sleep or keep awake at the wrong time. Now, what we can do in a laboratory setting is to experimentally examine the effects of instances like shift work and jet lag by artificially inducing a misalignment between the circadian system and our sleep-wake cycle. And this is the type of protocol that I have conducted, and I will show you some of the behavioral data from this protocol. So, there are quite a few different ways in which you can induce a misalignment between the circadian system and the sleep-wake cycle, but the cleanest and the most accurate way of doing this is to use something called a forced desynchrony protocol. And let me take a few minutes to actually go through this protocol. So in this protocol, what you effectively do is put people on a very long sleep-wake cycle. So for example, you put people on a 28-hour day where the wake is 18.67 hours long, and the bedtime is a little more than 9 hours. But the key in this protocol is to maintain this sleep-wake cycle over an extended period of time. So what did we do in this particular study? We had a 10-day protocol, which was 28 hours in length. Each waking day was 28 hours in length. But the key was the bedtime on each day started 4 hours later than the previous day, as you can see in this figure. So you have wake period 2, where the bedtime started at midnight, wake period 3, it started at 4 a.m., so on and so forth, until at the end of the protocol, they returned to a midnight wake time. And if you look at this protocol, somewhere around day 5, which is wake period 5, you can see that the sleep-wake cycle is completely out of alignment, completely 12 hours out of alignment with their habitual sleep-wake time. So effectively, in this type of a protocol, What we can do, or what we achieved to do, was send someone virtually around the world in 7 28-hour days, 4 time zones at a time. So you can now imagine the jet lag and the desynchrony between the sleep-wake cycle and the circadian system. Of course, this is not the only thing you have to do. There are a few other environmental conditions that have to be maintained in this protocol. You have to keep people in a time-free environment, so they effectively cannot know what the time is outside the laboratory. So the human circadian clock is very, very adept at picking up time cues or using all kinds of time cues to maintain a synchrony between the sleep-wake cycle and the circadian system. So the first thing you do is you keep people in literally a windowless room, so they have no idea of the light-dark cycle outside. The other thing that you have to do is to maintain people in dim light, so that the circadian system of each person runs at its normal period and doesn't adjust to a light-dark cycle. So maintaining people in dim light effectively keeps their circadian system desynchronized from the sleep-wake cycle, and is not able to use the light cue to resynchronize. So it's a tricky protocol that requires many different conditions, but I guess this has been done many, many times. So how can we go about getting data that allows us to look at the circadian regulation in a meaningful way? The first thing that you need is you need a circadian marker, a physiological circadian marker. And a very standard marker is the melatonin rhythm, because as I will show you in the data a little bit later, it is an extremely robust marker and shows beautiful circadian rhythms. So in this particular protocol, we collected hourly blood samples during 3 cycles of the protocol. The beginning of the protocol, where the melatonin rhythm occurred, was synchronized to a normal sleep-wake cycle, as you can see over here in the top red profile. Melatonin levels were high at night and low during the day. Participants were sleeping at night and keeping awake during the day. We also collected melatonin hourly blood samples on wake period 6. This is where the sleep-wake cycle was completely out of synchrony with the circadian system. And what you can see, and I hope you observe, is the melatonin rhythm still occurs at the same time as it did on the habitual sleep-wake cycle, except that here participants are trying to keep awake when the melatonin levels are high and sleep when the melatonin levels are low. So this is where you can see a desynchrony between the two biological oscillators. And then we again did blood samples on the very last day when the system resynchronized, when participants were sleeping at their habitual sleep-wake cycle. So those were the 3 points that we collected 28-hour melatonin samples. And what this did was allowed us to estimate a time point of interest in the melatonin cycle, and we used that particular time point as the circadian clock anchor. In this study that in the data that I'm going to show you, we use the melatonin onset. This is the time when melatonin levels reach 25% of the daily maximum as our anchor point. You may find other studies in the literature that use the melatonin peak or offset as anchors too. They're all fine. The only key thing is to find a time point in the circadian clock that can be used as an anchor. And again, this is not the time point of the clock in the brain. It's an indirect measurement. It's a time point in the circadian rhythm of a physiological hormone. Now, What we also did was to administer several different cognitive tasks that I have outlined over here in the left at regular intervals during the waking episodes. And in the forced desynchrony protocol figure, you can see these as blue squares. So it was roughly at 2-hour intervals that we administered all of these tasks. So what we were doing throughout the protocol, if you can see this, is we were sampling cognitive capacity and cognitive functions right through the circadian cycle, spanning the entire protocol and different times awake. And this is kind of the beauty of the circadian protocol. What you can do is you can sample data points right throughout the 24-hour circadian cycle of a person, and you can sample them at different times awake. So effectively, you can get an independent view of the circadian influence on cognition and an independent estimate of the time awake influence on cognition, which is a proxy for the sleep homeostat. And you can get the interaction between, you can get the interactive influence between these two factors as well. And I will show you this as we go through the data slides. And then, of course, you can use the, you can then, for your statistical analyses, align the time points either to the circadian marker or to the sleep homeostatic marker. So how do you do this? For the circadian marker, you can align the data point to the onset of the melatonin rhythm in this case. You can align it to the offset of the melatonin rhythm or the peak of the melatonin rhythm. And by this, I mean you have a time point at which each data point was collected. You simply calculate the lag between the time of the data point and the time of the melatonin onset, which is the same throughout the protocol because your melatonin rhythm just goes through its 24-hour cycle. For your time awake or sleep homeostatic level, that's a bit more simple. It's simply the difference between the wake point on a particular wake episode and the time at which the data point was collected. And then you can apply your magic of statistics, do all kinds of analyses. With this particular data set, we used two different kinds of analyses. We did a linear mixed model ANOVA, where circadian phase, time awake, and sex were fixed factors, and subject was a random factor. And we also used total sleep time as a covariate, because it is important to remember that both sleep and how you sleep, as well as your time awake and circadian phase, all interact to influence your cognitive function or behavior. And in this case, because people went through their sleep-wake cycle at different sleep phases, we didn't know whether participants were sleeping the same amount throughout the protocol, so we used that as a covariate. Of course, we also used a nonlinear model, and this is simply to get the amplitude of the circadian rhythm and the slope of time awake, and you will see this data a little bit later on. Now, before I kind of really dive into the data, I'm going to talk you through a little bit in detail about how you can visualize the data from a forced desynchrony protocol. So on this figure over here is a visualization of the circadian effect. And now the two images that you see are really mirror images of each other. It's nice to plot it this way so you get a sense of the circadian rhythm. So what do we have in this plot? On the bottom is the circadian phase in degrees. 0 degrees represents the time of the melatonin onset. Now, in this particular data set, on average, the melatonin onset for our participants occurred at 2200 hours. So on the top x-axis, you can see the relative clock time. So 0 degrees, which is when the melatonin onset occurred, was at 2200 hours. 120 degrees then is 6 a.m., 240 degrees is noon, and then that is again repeated in the next mirror image of the figure. On the x-axis is just your cognitive or behavioral data point. In this case, it is the Karolinska Sleepiness Scale. The purple area plot is the average melatonin rhythm of all the participants in our study. And this is kind of nice to plot because you can see the melatonin onset occurring at 2200 hours. You can see where the melatonin levels peak, and effectively, you can get a sense of the biological night, which is where you see the high melatonin levels in the area, and then you can see the melatonin levels go down, and you can see the biological day. So you get a nice sense of the rhythm. And this, of course, matches up with the clock time as it should. Now, the red data points simply represent the sleepiness scale in this instance. And what is nice to observe is that sleepiness levels were really high during the night and they were low during the day. Or as indicated over here, performance was really poor during the night and performance was relatively good during the day. I also want you all to note that between 12 and 6 p.m. is when you see really high levels of performance, and that is what we call the wake maintenance zone. It is when your alertness rhythm really peaks right before you go to bed. Likewise, performance levels were really low during 6 a.m. or sleepiness was highest during 6 a.m. Again, it matches up with what is known about this system and how it operates. Over the decades of research, this is when you are the sleepiest. And in fact, this is when, and perhaps I am repeating cliches, this is when a lot of the industrial accidents, the big ones, like the Bhopal gas tragedy and so on and so forth, they all occurred around between 6 and 4 a.m. in the morning, and they all occurred due to human error. And you can see this from a very circadian perspective, how that could contribute to it. Now, the time awake effect, of course, is a little bit easier to understand and visualize. You simply plot time awake on the x-axis as hours or minutes, and the y-axis is the data point. Again, poor performance goes from top to bottom, and what you can see is with every waking minute, your performance goes down. You are not that good towards the end of the day, especially if you kept awake for 18 hours, and you are also very sleepy. How can we visualize the interaction of these two? And I apologize, this is a complicated figure. Again, the left and right are simply mirror images of each other, but it's good to visualize the rhythm. So let me explain this figure. This is how one can visualize the interaction between the circadian system and the sleep-wake homeostat. The bottom x-axis is circadian phase in degrees. And in this particular data set that I'm going to show you, 0 degrees, which is the onset of the melatonin rhythm, occurred at 2200 hours. The top x-axis represents relative clock time relative to the circadian phase in degrees. The y-axis now represents time since awakening on the protocol. So it's simply a march from wake time to sleep time on a particular wake episode in the protocol. And what you can see over here is your day, and these are contour plots of the data. So when you have been awake for 16 hours, your performance at the circadian nadir is really, really poor. You are really shot to pieces over there. In contrast, if you've just been awake for about 2 hours at your circadian nadir, you're not doing that bad. You're okay. You're still performing not too great, but you're okay. The basic point is your performance at the circadian nadir, or your deterioration in performance at the circadian nadir, is amplified with time awake. And this is why night shift work happens to be such a big problem. Most people on night shift are not able to sleep very well during the day. And so they are typically awake for over 24 hours by the time they get to the middle or end of their shift. And that's when they really, really start to feel the effects, and performance goes down, errors happen, injuries and accidents happen. And this kind of illustrates the point very well. So in our particular protocol, what did we find? And what I'm going to show over here is a quantification of the circadian and time awake effects independently, and the circadian by time awake interaction on cognition. And these are simply effect sizes. It kind of illustrates all the measures that we did, and you can kind of get a global picture of it. So what is the takeaway point from this figure? What you can see in this figure is that the effects of circadian phase and time awake are really more pronounced for subjective measures of sleepiness and task demand than for many measures of attention and working memory, than for the more objective measures. And do keep this in mind. This is something I will allude to a little bit later on. That's just what we found. Subjective measures seem to be more sensitive to these manipulations than objective measures. And if you have any questions about that, we can discuss this in the question time. The largest circadian effects were observed again for sleepiness, mood, and subjective effort, whereas the circadian effects on objective measures of attention, working memory, and temporal processing were much smaller. The data are what they are. However, I always like to show this slide, because this is what got me started on sex differences. And this kind of summarizes the data that I've struggled with since I was a postdoc, which is now over 10 years, I think. So the first time I ran this study, I was very excited to run a new cool visual task. It was a visual search task. Only the melatonin rhythm was nice and rhythmic and showed a circadian effect. I did not find anything on this cool task, bummer. I then ran a simple reaction time task, which was the psychomotor vigilance task. Yeah, there was a beautiful circadian effect. At that time, I was, I came from a cognitive background. I was kind of a, snotty cognitive psychologist, and I really didn't care about simple reaction time tasks. Hence the statement, I don't really care about this task. And to add insult to injury, I found that a self-report scale, a 9-point Likert scale, showed an even better circadian rhythm in data. Again, cognitive psychologists do not like self-reported measures very much. They go for the really objective measures that they think somehow reflect what happens in the brain. But here was the data. I looked at a new derived measure, which was D prime, found nothing. Looked at a different response time, found a nice rhythm. And then it was just like, what? I don't understand it. These things are very different. They're not consistent. Anyway, going forward, I had the protocol 1/3 done. I found 5 subjects, found a beautiful circadian rhythm. Protocol halfway done, 10 subjects, circadian rhythm in the cognitive performance disappeared. But then again, every single time I found a rhythm in melatonin. Yep, at that point, I hated melatonin. So the question was, what was going on? And this was something I encountered over and over again. And of course, part of the argument is that you use different tasks, you use different scales. They are all differentially sensitive to circadian and sleep challenges. And indeed, you are going to find these differences, different protocols, you're running them in different labs that could also contribute to it. True, but I still felt there was something missing, and it was kind of half a story, not the full story. Because what you want is to say, are there actually any physiological factors or brain factors that could be contributing to this difference? And of course, looking through the literature, and at that time, sex differences were not a popular topic, but still looking through the literature, people had reported sex differences in human circadian and sleep characteristics. So for example, there is an earlier timing of clock gene rhythms in the brain in women. Women have a shorter intrinsic circadian period of body temperature and melatonin rhythms. They have an earlier timing and a larger amplitude of the melatonin rhythm. And there is an earlier timing, longer sleep, and more slow wave sleep in women. So over the years, people did report sex differences in the physiology of sleep and circadian rhythms. So in this particular data set, we decided to ask the question, are there similar sex differences in the cognitive and behavioral measures? Now, I did see that over the course of this semester, you've had a lot of talk in sex differences, so probably you are aware of this. There is a great complexity in defining sex, and I think this slide highlights it. You have the biological sex differences, and then you have the sociological, anthropological, and psychological sex differences, which you may call gender, if you will. At the time we analyzed the data, we just took a very simplistic, rather naive approach and looked at sex differences as defined by biological sex, and only looked at male and female differences and not intersex differences. Of course, since the data were published, I've delved a lot more into this literature and realized the complexity of the whole area, and realized a lot remains to be done. Nonetheless, I will now show you some of the data we have on this very simplistic difference, if you will. So we first decided to see whether we could replicate the sex differences in melatonin. And if you recall, we collected the melatonin data during 3 points in the protocol. The circadian rhythm within each participant was exactly the same throughout the protocol as it should have been. And here are the data. The data points on the top is the melatonin profile of women shown in red and men shown in purple. And as you can see, we replicated the finding of a higher amplitude in the melatonin rhythm in women. Relative to men, we did not find any significant timing differences. And at the bottom is a box plot showing the melatonin levels. And again, the same difference. Women have a higher melatonin amplitude and higher melatonin levels relative to men in our young sample. What about sex differences in sleep? And I will go through these data slides. The first data on the left shows percentage of non-REM sleep, percentage of REM sleep, and percentage of sleep efficiency. And this is the circadian effect. The next one shows percentage of non-REM sleep, REM sleep, and sleep efficiency as a function of thirds of the sleep episode, the first third and last third of the sleep episode. In the right over here, we show slow wave activity in the frontal, central, and posterior areas. And again, this as a function of circadian phase and thirds of the sleep episode. What we found in our analyses was that slow wave activity was significantly different between the sexes, showing a lower amplitude in women over the posterior and central regions. So This is basically what we found. Sorry, a higher amplitude. That was not lower, that's a higher amplitude. Again, mirroring what was found in the literature. But what about the circadian modulation in cognition, which is the main topic of interest over here? So from our nonlinear analyses, we were able to extract the amplitude of the circadian rhythm in various cognitive measures. And if, as you can see over here, the amplitudes are represented in red for women and purple for men, and these are the average amplitudes of the data set with the standard errors. And what you can see is that the circadian modulation in cognition in a number of these cognitive measures was larger in our female subjects than in our male subjects. And if you plot the data in a different way, if you just plot the circadian rhythm in these measures, you can kind of see the same thing. Overall, the amplitude of the circadian rhythm is much more pronounced in women than in men, such that performance during the early morning hours was more impaired in women than in men. And by early morning hours, it really reflects the biological night or the circadian sleep promotion zone. What about the effect of the sleep drive? Here, we did not find any significant differences between men and women in all of the measures. And you can see this in the slope of the time awake effect on our measures, as well as the data points plotted as a function of time awake. As a function of time awake. There was really no significant difference. And even in places where you see a difference in the intercept, the slope was really not different between men and women, showing that the decline in performance with time awake seemed to be similar between men and women. So to summarize the findings I've shown so far, the circadian rhythm in cognition seems to be larger in women than in men, at least in our sample of young subjects. Now, the impact of the sleep drive or the time awake effect seems to be very similar between women and men. The decline in performance with time awake was the same in our sample of subjects. However, there is an open issue that we were not able to address in this protocol, and a standard question that I get asked everywhere. What about menstrual phase? Because that's a very big biological difference between men and women. Now, there is certainly emerging data in the literature showing that cognitive domains like attention, emotion, response to reward, and spatial performance are all affected by menstrual phase. So one may then hypothesize that alertness would be influenced by menstrual phase, or these cognitive domains would be influenced by menstrual phase, given that progesterone is released during the luteal phase and seems to mitigate the effects of sleep deprivation on cognitive performance in women, as shown in some of these studies. So the prediction then is that in the follicular phase, women will show greater sensitivity to the effects of acute sleep deprivation or circadian misalignment in cognition. And there is very nice data by a paper by Vidafer et al., which shows attentional lapses on the left over here and extremely long attentional lapses on the, extremely long attentional lapses on the right. And I hope you all can see the really dramatic difference that cognitive vulnerability due to sleep deprivation is indeed more pronounced in women during their follicular phase. So in both of these figures, the follicular phase is shown as red symbols, the luteal phase as blue symbols, and men in gray. So really, in the luteal phase, this difference seems to disappear between men and women. What about physiology? We measured the melatonin amplitude in men and women. Another very robust physiological rhythm is temperature. Sorry. And indeed, again, Vidoffer et al. In their data set found differences in the temperature rhythm between the follicular and luteal phase. Of course, in our sample, we were not able to look at this difference because we didn't collect the data and we didn't run the study to look at this data. But the temperature rhythm shows this difference. And this may give a better handle or a sense about the underlying biology, the finer resolution of how these differences are realized in the brain and the body. So in conclusion, at least what I have learned so far is that there is a diurnal rhythm, a day-night rhythm in waking performance that is modulated by circadian phase and the sleep-wake homeostat. All of us will experience this across the board, but they vary across cognitive domains and between individuals. It seems to be the case that when you challenge the system by either inducing circadian misalignment or sleep deprivation, your subjective response or your subjective judgment of how you are performing seems to be more sensitive than your objective measures. And of course, one factor driving these individual differences is sex differences. The circadian amplitude, at least in many cognitive measures, is stronger in women than in men, but this seems to be modulated by menstrual phase. In other words, women seem to be more susceptible to cognitive impairment arising from circadian misalignment and extreme sleep deprivation during the follicular phase. And of course, these findings have implications for the real world, shift work and extended work schedules, but this is a very, very small part of the story. As I showed in that slide, the definition of biological sex itself is very complex. There are other physiological factors, for example, age differences. Children and adolescents may exhibit these things much more differently. And of course, something that people are probably just beginning to realize and should realize is that our circadian system is, of course, influenced by the light-dark cycle. You cannot really separate out the influence of light on all of this. But the light-dark exposure that we have on a daily basis varies vastly around the world. And most of these studies are run in a very small part of the world in Western industrialized societies. Whether these data and findings apply to the larger diversity one sees in human population is a very open question. And I would like to acknowledge people at the Surrey Sleep Center where the data that I presented was collected. And thank you very much for your attention.